applying of high amount of auxins. So by the applying of high amount of auxins, so it leads to be development of roots from the explant or callus, that is called rhizogenesis. At the same time, a high amount of cytokinins and a less amount of auxins applied to the callus or explant. So it leads to be growth of shoot, that is called collagenesis. So rhizogenesis, high amount of auxins and less amount of cytokinins. So roots develops from the explant, that is called rhizogenesis. In collagenesis, high amount of cytokinins, less amount of auxins. So in that time, development of shoot system from the explant, that is called collagenesis. Now the next callus. So callus. So any part of tissue which is extracted from the plant, that is called explant. Now these explant taken onto the nutrient culture media. So in that culture media, these explant cells undergoes to many divisions at the short time and produces a mass of cells. Now these cells are called callus. Next, organogenesis. By the production of shoots, roots, floral buds from explant or callus, that is called organogenesis. So these organs such as shoot, root or floral buds, which are directly develops from explant, that is called direct organogenesis. Or these parts such as shoot, root or floral bud, so develops from callus, that is called indirect organogenesis. So it means based on the type of development, so these organogenesis, which is classified into two types, direct organogenesis and indirect organogenesis. If these parts or organs develops from explant, that is direct organogenesis, or these plants develops from callus, so that is indirect organogenesis. Now the next one, somatic embryogenesis or synthetic seeds. So when the explant taken into the nutrient media, now these explant cells repeatedly divisions and produces mass of cells, that is called callus. So now these callus cells encapsulated by sodium alginate or sodium hypochlorite. Now it is called synthetic seeds. So it means synthetic seed is nothing but somatic embryo, vegetative cells which are used as a synthetic seeds. Now next, encapsulation of embryos with hydrogels. So somatic embryos are encapsulated by different methods. So these encapsulation with proper materials and the structure will not only protect the embryos from physical damage or desiccation during the delivery or the sowing process in a greenhouse. So the somatic embryos induced in appropriate nutrient mediums, and these are mixed with sodium alginate. Now these embryos loaded alginate gels, and these alginate containing embryos dropped into a solution, that is calcium chloride. Now the surface complexion begins immediately, and each droplet forms calcium alginate capsules around each embryo. Now this is called somatic embryo. Sakamoto in the year of 1992 improved the quality of hydrogels and developed a self-splitting or self-breaking gel. Further rinsing the beds thoroughly with soft water or tap water for 40 to 45 minutes, and these embryos immersing them in a monovalent caution solution of potassium nitrate for 60 beds. And entire this process with 6 hours in humid conditions. So now these synthetic seeds lack an endosperm. So these synthetic seeds lack an endosperm. Why? Because it is not participate in sexual reproduction. So synthetic seed, it is nothing but it is a vegetative cell which is formed encapsulated by a chemical compounds. Now this is called synthetic seed. Now the next one, androgenesis, gynogenesis. So andros, it means male, gynous, it means female. So in that androgenesis, formation of microspores from plantlets or callus by the process of pollen culture, that is called androgenesis. Shipragruha Mukherjee and Maheshwari, Department of Botany, University of Delhi. So these both scientists reported direct development of embryoids and plantlets from microspores of the Datura inoxia. Now the next one, gynogenesis. In vitro regeneration of haploid plantlets from unpollinated female gametophyte by culturing ovules. So haploid plantlets developed from unpollinated female gametophyte. So unpollinated female gametophyte. So this female gametophyte is nothing but embryo sac. By culturing ovules, that is called gynogenesis. San Nom 1976 reported that haploid plantlet regeneration via gynogenesis in barley. So androgenesis reported by the scientist Shipragruha and Maheshwari, the plant Datura inoxia, and gynogenesis San Neom reported in the plant of barley. Now the next, Miller and Skoog experiments. The tissue culture process, we are used nutrient culture medium, that is MS medium, that is Murashige and Skoog medium. You see, in 1957, Miller and Skoog demonstrated plant growth regulators controlled bud formation in tobacco stem segments by varying the concentration of auxins and cytokinins. So that is Miller and Skoog experiment. So in this chapter, so this terminology, this very, very important in that totipotency, rhizogenesis, collagenesis, xylogenesis, androgenesis, and gynogenesis and organogenesis. So these keywords are very, very important terminology and tissue culture. Now the next one, types of tissue culture. In the unit 5, types of tissue culture. So there are different type of plant tissue culture techniques are there. So these different tissue culture techniques based on the explant used. So in that, cell culture, organ culture, callus culture, protoplast culture. So that's cell culture, organ. Cell, it is a single living organelle that is called cell. Second one, organ. Third one, callus. Organ, it is formed, organ is nothing but a group of tissues which are joined and forming a structure that is called organ. Now the next one, callus. Callus is nothing but a group of cells. And the last one, protoplast. Protoplast. So there are protoplast and protoplasm. So these two words which are different, so both are present in a plant cell. Protoplasm, nucleoplasm plus cytoplasm. So that is together called protoplasm. Protoplast, cytoplasm associated with plasma membrane, that is called protoplast. So these two terminologies different, protoplast and plasm. So that first, cell culture. Cell culture is the culture of isolated individual cells. In the isolated individual cells. So it means a group of cells or a single cell which is extracted from the selected plant. Now that cell obtained from an explant tissue or callus. So that is regarded as cell culture. Now these cell cultures are carried out in a dispension medium and are referred to as cell suspension culture. That next, organ culture. Organ culture is isolated, select a plant organ that is like embryo, seed, or root, endosperm, anther, ovule, ovary, meristem, or nucellus. So these are act as organs. Callus culture involves the culture of differentiated tissues from explant, so which differentiates in vitro to form callus. Now the protoplast culture. So it is the culture of plant protoplasts in the laboratory. So in this unit, cell culture, callus culture, and protoplast culture is explained. See in the first one, cell culture. Cell culture. So during the cell culture, Hebert Land first proposed and first observed this cell culture by the in vitro. So in this cell culture, isolation of single cells So it means isolation. So selection of single cells from explant. The next cell culture techniques. So there are two cultures are there. One is the batch culture. The second one, continuous culture. See, this is the diagrammatic structure of suspension culture. So in that first, that is explant taken in the nutrient culture media and closed with cotton. Entire the explant with the culture media that is closed with cotton. The next second image, it indicates agitation in a liquid medium. Now in this liquid medium, selection of a single cell. Now the cell transfer into the micro chamber culture. So during the micro chamber culture, the cell repeatedly divides and produce a mass of cells. So it means formation of callus in a micro chamber culture. Now the next, these callus transferred into another nutrient culture media. So in this nutrient culture media, these callus by the application of phytohormones such as auxins and cytokinins in a different concentration. Now the callus cells undergoes to divide and produces organs, and these organs further develop into new plantlets. Now these formation of new plantlet transferred into another parts. Now this entire plant acclimatization to the natural environment. So this is the process of cell suspension culture. So in this culture, first of all, we have to take an explant from the selected plant. Now the selected plant explant taken into the nutrient culture media and closed with cotton. Then next, these explant, after some time, these explant transferred into a liquid media. For agitation. So in the liquid medium, selection of cell, single cell, it is easy to place it into the liquid media. Now these cell transferred into a micro chamber culture. In that time, these cell divides and produces mass of cells in the micro chamber culture. Later, these callus transferred into a basal media by the application of phytohormones such as cytokinins and auxins in different ratios. So in that time, the cytokinins concentration is very high when compared to the auxins. So that time shoot develops from the callus. Then next, auxin's concentration is applied to very high, and cytokinin's concentrations is very low. In that time, roots which are formed from the explant. So after a period of time, so these cultured plant pots, roots and shoots which are associated and forming a complete plantlet. The next, these plantlet transport into another pot for the culturing of. So these plantlet transport into another pot, and these potted plantlet acclimatized to the natural environment. So this is a suspension culture process. So next, see in the next image, this is callus culture. Already you know callus is a mass of cells. So that cells are developed from the explant. So in this callus culture, first formation of first taken explant, any part of plant which can be taken and used as a explant. So these explant tissue which is extracted from the natural environment. So it means these explant to be sterilized with the autoclave. After sterilized the explant, these explant is taken into the nutrient culture media. That culture media, that is agar media, agar medium. So agar, by the application of agar into the liquid media, now it is solidified and forming a humus-like structure. So solid medium that is formed by the application of agar. So these agar medium having agar and nutrients and phytohormones and distilled water. So that is solid media. So the solid media having agar medium, a liquid media. So there are two types of medias. One is the solid medium and the second one liquid media. So liquid medium contains distilled water, micronutrients, macronutrients, phyto vitamins, amino acids, and sugars. So these all nutrients mixed in distilled water, that is called liquid media. So it means liquid medium do not having agar. If the agar is applied, that medium is called solid media. And one of the another media is present, that is solid media mixed with phytohormones, that is called basal medium. So see in this callus culture, sterilized explant taken into the agar media. Now these explant undergoes repeated divisions and forming a mass of cells. Why? Because these nutrient medium, agar medium having rich in nutrients and water, vitamins and amino acids. The next, these medium placed on the lighting area. So in that time, these explant cells absorbs water and minerals from the medium and undergoes repeated divisions and forming a mass of cells. Now these mass of cells, that is called callus. The next, these callus subculturing of new medium. So a new medium is nothing but by the application of phytohormones in a different concentration. Later, these callus taken into the liquid medium. So liquid medium does not having agar agar. So this is a process of callus culture. Now the next one, see, this is the steps involved in protoplast isolation culture and regeneration. So in that first image that indicates potted plant, it means parent plant. Now the second image, leaf sterilization, it means explant, the explant that is taken, that is leaf. So now these leaf sterilized by the application of different chemicals. The next, third image, epidermis peeling. On the fourth image, leaf segments in enzyme mixture. The next, removal of enzyme solution. Next, suspending enzyme digested material in washing medium. Next, pellet after centrifugation. The next, cell debris. Next, isolated protoplast, wall regeneration, clumps of cells, callus tissue from protoplast, differentiation of shoots in callus tissue. The next, new plantlet that is formed from protoplast. So these are different steps. So in that entire the process, first we have to isolation of protoplast. Now the isolation of protoplast, there are two methods are present to be taken protoplast. One is the mechanical method, second one enzymatic method. That mechanical method, isolation of very small of protoplast. The enzymatic method, high amount of protoplast should be extracted from the enzymatic method. Now these protoplast taken from leaves or roots or shoot apices, fruits, embryos and microspores. Among these, the mesophyll tissue of fully expanded leaf of young plants or new shoots are mostly frequently used. Now the advantage of enzymatic method, so it includes good yield of viable cells. Now the next one, isolation of protoplast from the leaves. So this protoplast taken from leaves after a period of time, this protoplast cultured by using of cultured media. So in that culture methods are, one is the feeder layer technique, co-culture of protoplast, and micro drop culture. So these are the different methods for using protoplast culture methods. So in that first one, feeder layer technique. So this feeder layer consisting of non-dividing living protoplasts. So in the feeder layer technique, protoplast which are taken onto the agar medium and the isolated protoplast are plated in a thin layer of liquid medium. In the co-culture of protoplast technique, by using of chemicals, so these chemicals which helps in the generation of cell wall and cell division. And third one, micro droplet technique. So in micro droplet technique, they are using caprock dishes with outer and inner chambers. The inner chambers carries several wells wherein the individual protoplast in droplets having nutrient medium. So these are the different cultured medium techniques. So in this entire chapter, one is the cell culture and the protoplast culture. So these are the important questions for the examination purpose. Next one, callus culture, suspension culture, protoplast culture. So these are also very important for our examination purpose. Next in organ culture process, already you know organ culture. So entire the tissue culture technique, root culture, shoot culture, leaf culture. So these are root, shoot, and leaf. These are vegetative parts in plant, and these vegetative organs taken onto the culture media and growing new plantlets. So that is organ culture. And the next one, reproductive culture. So in the reproductive culture, the formation of male female reproductive organs such as androecium and gynoecium by a process of androgenesis and gynogenesis. So that is a reproductive culture. So it means entire organ culture that is classified into two types. One is the vegetative organ culture, second one reproductive organ culture. That vegetative organ culture, so that is further divided in different types: root culture, shoot culture, leaf culture or node culture. So these are all vegetative organs from a leaf, from a plant. Reproductive organs such as root and shoot. Sorry. Reproductive organs such as androecium and gynoecium. So androecium produces male sexual organ that is called microspores. Gynoecium produces female sexual organs that is embryo sac. So embryo sac act as a female gametophyte and microspores act as a male gametophyte. By the production of male gametes by a process that is called androgenesis in a tissue culture, the production of female gametophytes by a process that is called gynogenesis. So this is organogenesis. So next block 3.